So just keep talking.
Okay.
All right.
I'm sorry, you guys.
Nakia, scoot over towards Nate a little bit so I'm gonna be in your face. All right. Hello everyone, how you doing, Dr. Faison? This is Group 1, talking about regulation of gene expression. My name is Malik Dockery.
My name is Nakia Lawrence.
My name is Nathaniel Plummer.
I'm Kaleo Lachide.
And we're going to get started. Let's share our screen with you guys to see the presentation. And let's begin.
Okay, so like Malik said earlier, we're going to be talking about regulation of gene expression. And before we go on, it's certain terms that we should all be able to know and understand before we go to any in-depth knowledge. So first of all, I'm going to be defining these terms. The gene is a DNA sequence that codes for a specific peptide. A genome is a complete set of genes in an organism. Gene expression is the expression of a gene that leads to the generation of a functional peptide. The key word here being functional, the peptide has to be functional. Transcription is the first step of gene expression that involves the copying of a particular segment of DNA into mRNA, and what does this is RNA polymerase. Translation is basically using the mRNA produced from that transcription to make proteins. So here the mRNA produced from the transcription is decoded by the ribosomes, and the ribosomes use that to produce a specific polypeptide. So this next slide talks about the levels of gene expression. So there are levels at which genes could be expressed. So there are 5 different control points of gene expression. We have the genome, the transcription, DNA processing, and nuclear export, which is post-transcription, translation, and post-translation. But generally, these last 3 steps are known as the post-transcriptional control mechanisms. So I'm going to start with the chromatin, which has to do the chromatin level of gene expression. The genome level of gene expression, which deals with the chromatin. So before we move on, we should know the definition of a chromatin. The chromatin is a material of which the chromosomes of organisms are composed. So basically, chromosomes are made up of chromatin in simple words. So as this right here, these are ways that genes could be expressed in the chromatin. Moving on to the next slide will be bigger so that we could all see that. So this right here is basically just giving you the organization of the chromosomes. So obviously the chromosomes have to be made up of something. The chromosomes are made up of chromatins, and how you make these chromatins, they're just basically condensed nucleosomes, and nucleosomes are basically histones and DNA. So the DNA is wrapped around histones and to get nucleosomes, and then when you further condense the nucleosomes, you get chromatins, and these chromatins make up the chromosomes. And there are ways to regulate gene expression, and in the genome level, I'm going to start with gene amplification and deletion. And basically, I would just start by saying that the cell tries to avoid any type of way that we would use energy that is not necessary, like basically using energy that on something you don't need. So let's say if my cells don't particularly need gene X, my cells don't see the need to make more of gene X. So that gene will be deleted. And similarly, if my cell needs more of a particular gene on a chromosome, we would need to amplify that gene. So that is basically gene amplification. So gene amplification, the word amplify generally means to make more of. And gene amplification is the case where you produce multiple copies of the same gene. This is done by replicating the DNA in a specific chromosomal region. And amplification of genes could sometimes mutate and then encode for different functional peptides. So when you keep amplifying a particular gene, which is basically making the exact same thing like of a particular gene, it could sometimes mutate where you have that mutation code for something else, code for a different peptide. And this is a way that you regulate gene expression, although this is very rare. The opposite, also gene deletion, is a very rare way of regulating gene expression. Moving to DNA rearrangement. This is also another rare method. DNA rearrangement involves the movement of DNA segments from one location to another. So sometimes DNA rearrangement of non-coding sequences can lead to the expression of a gene. And I believe Malik will be talking to you about that later. Also, rearrangement of a gene can also cause an enhancer and a promoter to come in close proximity so that transcription is initiated. So in a couple of slides, I would explain to you, well, generally where these enhancers, where the promoter is located in a gene and how far apart they are from each other. And basically, when you arrange DNA, you could arrange it in such a way that the enhancer and the promoter region could come close to each other. And when this happens, you could initiate transcription, because then when the enhancer binds to the promoter region, it makes it noticeable for RNA polymerase to locate the promoter region and then bind to it so that you could start transcription. Also, DNA rearrangement creates different antibody genes with a mechanism involving enhancers. So the next one is going to be decondensation and condensation of chromatin. I generally would put that together with the fourth one, which is histone modification and chromatin remodeling, because when you methylate or when you add an acetyl group, depending on what the functions are, as we will see in a few minutes, they could cause the chromatin to decondense or condense. Condense meaning get them packed together. Decondense is getting them loose, the chromatin loosely away from each other. So you need to know that there are two types of chromatin. The heterochromatin, which is the condensed form, and the condensed form is usually transcriptionally active. And I say this because when they are condensed, when they are packed together, the promoter region is not necessarily noticeable to RNA polymerase in this way. And if RNA polymerase doesn't bind to the promoter region, it is not possible for transcription to occur. So that's why the condensed form is transcriptionally inactive. The euchromatin is the decondensed form, which is the transcriptionally active, because when they're decondensed, they're loosened up. So it is easy for identification of the promoter region which causes initiation of transcription. And histone modification, we know that histones are proteins found in chromatin around which DNA are wrapped. Acetylation and methylation are ways to modify histones. So acetylation is the addition of an acetyl group. Methylation is the addition of a methyl group. However, adding these two things have the opposite impact. And I say this because when you add a methyl group to the histones, it becomes inactive. But when you add an acetyl group to histones, they become active. So when you add these groups, they generally help to change the structure and activity so that the histones are, well, could park more around or less, as the case may be, the regions that they are surrounded to. So normally chromatin is condensed. We can see this in this picture. They are normally condensed. So a way to regulate gene expression is that when they are condensed and tightly packed, you can add an acetyl group, and this will help to decondense the chromatin and make it more loose, and it becomes transcriptionally active. So when you add the acetyl, you're loosening up the chromatin. Chromatin, you can identify your promoter region, it could bind, and transcription could occur. Histones are acetylated through HATs, and HATs are enzymes called, they stand for histone acetyl transferases. And when this happens, the chromatin is loose, like I said, the promoter is accessible to the RNA polymerase, and you could bind. Histone deacetylase is an enzyme that helps to recondense the chromatin structure. And in this state, RNA polymerase will not be able to bind to the promoter, and transcription of the DNA sequence will not be possible. So why is this useful? How does this help to regulate gene expression, basically? So if you want to, let's say we want to turn off, we don't want a particular gene to be expressed. So that way we could hinder transcription from occurring. And when we do this, we could do this by adding an acetyl group. And when you add the acetyl group, we do this by removing the acetyl group. And when you make the decondensed structure to be condensed. So this way, the transcription will be inactive, and then we won't get that gene to be expressed. But if we want more gene to be expressed, you can add acetyl group to this, and more of the gene will be expressed, transcription will. So this is just the way gene expression is regulated. Also, like I said earlier, methylation of histones work in the opposite manner. When you add a methyl group, the chromatin becomes condensed. So when it's condensed, the promoter region is inaccessible, and it is transcriptionally inactive, whereas unmethylation of histones cause a decondensed form of the chromatin and a transcriptionally active site. So over here is just showing that acetylation occurs at the C-terminal portion, and they both cause changes in the shape. And HDAC is the enzyme that is responsible for deacetylation, which is histone deacetylase. The enzyme responsible for acetylation is histone acetyl transferases. And this is just the basic summary of what I just said. This diagram right here basically shows the condensed form of a nucleosome, and basically if it's condensed, the activator cannot bind to the promoter, and if the activator cannot bind to the promoter, transcription is not going to occur. So what happens is the histone acetyl transferase, like I said, was the enzyme that makes that's responsible for acetylation. It binds with the activator, it binds to the nucleosome, and it decondenses this condensed nucleosomes, and it makes the promoter region to be noticed, so the activator could then bind, and transcription could occur. So chromatin remodeling is basically from the word is remodeling the chromatin, the structure of the chromatin using chromatin remodeling complexes that help to slide or expose the chromatin structure, the nucleosome structure, so that the promoter region is seen and the activator can bind to the promoter and initiate transcription. So this is what this diagram over here is basically talking about. Next is just basically everything I talked about is in activate transcriptionally active and transcriptionally inactive. Transcriptionally active is loose, as you can see here. Transcriptionally inactive is condensed, it's the condensed form. When it's active, the promoter region can be noticed and transcription could occur. When it's inactive, you cannot find the promoter region. And then right here you have the HAT, which is helps to decondense this structure, and the HDAC, which is the enzyme responsible for deacetylation. And then the next level of gene expression is transcription. And there are ways to regulate gene expression in the transcription level, in the initiation of transcription, which means like before transcription starts, how could you regulate whether or not these transcription could occur and these genes could be expressed. So we have the components of a eukaryotic found on a eukaryotic gene. The introns, the exons, promoter region, transcription start site, and the enhancer. So the exons, okay, so this diagram over here just shows all the parts of a eukaryotic gene. I'm going to explain in details what these parts do. So as we will learn in future, but just to be to understand this part, the intron is the part that is sliced out, is the non-coding part of a gene. So it is basically going to be spliced out. But the exon is the coding section of a gene. So that's what we have on this site. The start of transcription is basically where transcription starts. We have the promoter. The promoter, we have the core promoter and we have the upstream promoters, which generally the core promoters are generally don't change between eukaryotic genes to another. And the most common example of a promoter is the TATA box core promoter. And it's called this because the sequence of the nucleotide is TATA. The core promoters are where all the transcriptions and regulatory proteins bind to. So in the core promoter right here, transcription factors or regulatory proteins will bind, and until this binding, when this binding occurs, that is when RNA polymerase could identify that region and initiate transcription. So transcription start site is the RNA where is the site where RNA polymerase 2 binds to the site for the process of transcription to begin. So how do we regulate gene expression in this whole thing? So we have enhancers and silencers. From the words, we could tell that enhancers enhance basically, and silencers inhibit gene expression. So enhancers are distal elements found further upstream of a gene. So well, actually it could be further upstream, it could be further downstream, but they are far away from the promoter region. And what happens is that for enhancers, special transcription factors, special transcription factors that are, in the case of enhancers, activators, bind to them. And when this happens, you form a loop. So right here, we can see a loop. So the loop is basically bending of the DNA. The DNA bending protein, so gene expression by making more. Now, the silencers act in the opposite way. The silencers are also found upstream, further upstream or downstream, but they are they bind to inhibitors, and these silencers, when they bind to inhibitors, it's still, everything still occurs, the bending still occurs, but and then when they bind to the promoter, it shuts the process of transcription. It does not allow transcription to occur. So that's basically what this this figure explains. Moving to this next figure. So this figure explains the combinatorial action of regulatory transcription factors. So here is just the regular, gene, and you see your promoter region, you see the control element where you could find where you could find the enhancer, and then where the regulatory transcription factors bind to. So the difference here is that okay, so you should know that the regulatory transcription factors could be different from gene to gene. So it's not the same. But in this picture, the regulatory transcription factor is able to bind to the enhancer, and when it binds to the enhancer, it enhances it. The gene albumin is transcribed at a high level. But in the brain cell nucleus, where it does not bind, it is transcribed. It's still transcribed, but it's transcribed at basal level, because it's still going to happen. Transcription is still going to happen without the enhancer, but it's just going to be transcribed at a basal low level. So that is it for the regulation of gene expression in initiation of transcription in genome level.
Okay, so now we're going to get to alternative splicing. So I want you all to know that there are different types of splicing. Alternative splicing. There is alternative promoter, alternative poly A site, skipped exons, and introns. So for alternative promoters, it basically means that it is a different start from transcription. For transcription. So if you know, or if you don't know, the ribosome must connect to the promoter, and then the promoter will continue down the gene. And once the promoter has stopped, or the promoter sometimes will either stop or it will skip, because the promoter can only continue moving forward. So if you had one gene and the promoter is continuing down if you have one extron or intron and the promoter is going down, that means that it would possibly skip, and that means that the poly A site will stop transcription. So for the picture that you all see for exon skipping, which is in this figure right here, but here's exon skipping. As you can see, this is the intron, and this orange exon skips the blue exons and connects to the orange exon. So this orange exon right here skips this exon and then connects to this one. That is one way of knowing alternative splicing and skipping in exons. In intron retention, it states that the exons did not get sliced out, so it stayed all together. You see in this figure right here. Then you have alternative 5' splicing site, so you can see that this orange orange exon has skipped the blue exon and connected to this orange exon. You can see that this orange exon right here has now connected to this blue exon and did not connect to pre-mRNA and create many new proteins. So for alternative splicing, you know that there's different type of factors of alternative splicing. We have proteins and RNA, which binds to splicing enhancer and splicing silencers. So for splicing enhancers, it helps us assemble the splicing machinery, and for splicing silencer, it prevents the splicing machinery, which means it makes it stop. Remember, keep in mind that they both act as individual splice sites. So splicing enhancer is an activator and splicing silencer is a repressor. As for humans, pre-mRNA splicing is very important because in humans we have little genes, but there are many different types of proteins. But also we produce so many genes that some genes will either create beneficial effective proteins or proteins that can cause diseases. As we continue moving down, an alternative splicing mRNA decides which exons are kept and which exons will be spliced out. Remember, a gene is first transcribed in pre-mRNA and in DNA, and they both have introns and exons. So at the bottom, you see we have the 5 cap prime and the 3 poly A tail. In this figure, I'm going to explain why the 5 cap prime and 3 prime poly A tail is important. So as you see, we have our gene, and you can see the intron and exon labeled in the figure, which is here, and the exon is here, and this is our gene. So once the gene has been transcribed, then it will create the pre-mRNA. So that means the primary RNA transcription, which is this. Then I want you all to know first that the blue labels are exons and the yellow labels are introns. Once the pre-mRNA has been created, then we begin to form a 5 prime cap of the mRNA. Then mRNA is exported because the protons are binded to the mRNA. So this is where you can see the export of the mRNA. Then we finally create our new protein, which is here. But don't forget that we still do have translation before we get to the protein. So this next figure is also imperative to know about alternative splicing, and I'm going to break this figure down to you all so you can understand what is being discussed. So as you can see in this figure, which involves alternative splicing, you can see the long strand gene at the top, which is this. This is our long strand gene. It has the DNA coding for IgM and it has the exon and the poly A site. So this is the IgM, this is poly A site, and these are the exons. So then transcription occurs. The poly A site was skipped. It went to the left. So what I mean is, once transcription had occurred, this poly A site was skipped and it went to this side, which is here on exon 4. Then the exon to the right continued to make exons until it got to the poly A site because it could no longer bend to the exon. So what I mean is this poly A site went here, and then it continued making then the exons continue until it was no longer able to bend to the next exons, which is here. No exons was able to connect to exon 4. However, on the left, however, on the left, because the poly A site was skipped, it continued to bind exons 5 and 6, which are here. Then integrate into the membrane. So this is now our new protein, and it was now able to integrate into this membrane down here. But the figure on the right was not able to create the full protein, so it was not able to integrate the membrane. So as you can see that it was stopped here, and it was no longer able to enter the membrane down here. So the nuclear export is complete. Then post-transcriptional is the export of mRNA and go through the cytoplasm. So the mRNA only recognizes splicing, 5' cap, and the poly A tail. So what I mean by um, there is a protein that is binded to the 5' cap, and proteins binded to a poly A tail. Then the proteins are the pores, and then the proteins leave the mRNA. The reason why important nuclear export is important in gene expression, because if the ribosomes do not connect to the gene, then the gene expression will not occur. Also, it's important that our bodies go through translation transcription, because if not, we will not be able to continue to make proteins. And we all know that our bodies need proteins. And we're going to move on to the next slide.
Hello. Okay, we're doing the gene expression. So it's translational and post-transcription, splicing, stability, localization, and small non-decoding RNAs. Post-translational, protein modification, carbohydrates, lipids, phosphorylation, hydrolyzation, methylation, and pressure protein. Okay, for this figure, it's the first step is dicer cleaves into the siRNA. The second is subsequently combined with RISC proteins to form an sRISC after degradation of 1 and 2 siRNA. So basically, it's when a cell encounters a double-strand RNA, dicer cleaves it into a siRNA. It is subsequently combined with the RISC proteins to form one sRISC. For the second part is after degradation of the one of the 2 siRNAs. Third part is strands. The remaining strand binds to the sRISC by complementary base pairing to enter a target mRNA molecule in the cytoplasm, or to a target DNA sequence in the nuclear, leading to slicing gene expression at either the translational or transcriptional level. Okay, subcellular location. Is a protein subcellular location is prediction involves a computational prediction where protein resides in the cell. basically, a study of protein subcellular location is important to include protein function. Or protein subcellular location prediction involves in complementary prediction, whereas a protein resides in a cell. Subcellular location determines the environment in which a protein operates. A subcellular location influences the protein function by controlling access to an available or all types of molecular interaction partners. Targeting signal information into a probabilistic framework allows the accurate prediction of mammalization protein subcellular location. Okay. Initial translation. Translation is a process in which cellular ribosomes create proteins. During initiation, ribosome assembles around a target mRNA. Translation of mRNA molecules by ribosomes occur in 3 stages: initiation, elongation, and termination. During initiation, the small ribosome subunit binds to a start of an mRNA sequence, transfers RNA molecules carrying amino acid method binds to what is called the start codon of an mRNA sequence. Each corresponding amino acid is added to the growing chain and linked via a bond called peptide bond. So elongation continues until all codons are read. Translation is the use of an mRNA-directed protein synthesis and subsequent post-translation processing of protein molecule. So some genes are responsible for production of other forms of RNA that play a role in translation, including transfer RNA and ribosomal RNA. So it's 4 steps. I have translation involves in 4 steps. Initiation, a small amount of subunits of ribosome binds at a 5' end of the mRNA molecule that moves into a 3-directional until it meets a start codon. The second is elongation, a subsequential codon of an mRNA molecule determine which tRNA molecules link an amino acid binds to mRNA. An enzyme peptide transfers links to amino acids together using peptide bonds. Third is termination. The translation is terminated when ribosomes reach 1 or more steps. Ribosomal complexes in eukaryotes and larger, more complicated than in prokaryotes. And the 4th is post-translation process of a protein. Okay, for the figure. So for this figure, the first step, it says microRNAs are produced by a multiple step processing in which the first step, microRNA genes first transcribe into primary transcript that folds into a hairpin loop. Second is expanded from nucleus, cleavers, pre-mRNA into a smaller hairpin RNA. And the third is exported from nucleus and cleaver by directed to release microRNA molecules that is 21 to 22 nucleotide long. The 4th is mRNA then joins RSC proteins to form mRSC, which is directed by its miRNA to messenger RNA containing sequence complementary. And then the 5th is mostly common multiple mRNAs bind to the same messenger RNA. And then for the 5B, occasionally mRNA is exactly complementary to the site within a particular messenger RNA, resulting in a messenger RNA degrading by mechanism similar to observed in sRNAs. Okay, for RNA stability and degradation, the RNA stability is the extent of which RNA molecule retains its structural integrity and resists degradation, and base catalyzed hydrolysis under chaining of VO or vitro conditions. Different steps. They could do directional stability, regulational stability, iron regulation, nonsense, medicated decay. And consistently, many arises from variants in RNA stability, with transcripts that are more or less abundant than predicted RNA expression. From histones, data are inferred to be more or less stable. Genes with the abundance that has medicated by changes in RNA stability by RNA decay factors. That's it.
And now we're going to move on to the post-translational modifications. So after the translation process, polypeptides are released from ribosomes. The polypeptides enter the post-translational modification process where they are modified so they can become mature functional proteins. The modification may aid in proper folding of the protein, increasing stability, and inactive and activating function of the protein. So let's look at a few types of modifications. First, I want to talk about phosphorylation. Phosphorylation basically is adding a phosphate group to protein, which helps regulate protein function. In the figure to the left, the protein kinase mediates phosphorylations at amino acid, which isn't shown in the figure. Amino acid side groups, which are thymine and thymidine. However, phosphatases reverses protein phosphorylation by removing phosphate groups. Shown here on the right side, you see that phosphorylation causes conformation change in the protein activation. You can see right here at the top that there is a phosphate group here. You can see that there is a conformation change. However, you can see that there is still a phosphate group here, but there is no conformation change, which shows that this protein function is inactive. So next, we're going to talk about glycylation, addition of sugar, which can be a carbohydrate, which acts as signals that lets other cells know where proteins are going, what they're going to do, basically their functions. And it basically lets the body know where this cell is going to be or whatever. Now we're going to talk about proteolases, which is cleaving of the peptide bond. When some proteins are synthesized, they are inactive form of cymogen. Proteases, enzymes break specific peptide bonds in order to activate protein. And also cleaving of peptide bonds helps the protein fold in the correct way. Here shown in the figure, you can see that the breaking of the chloride and nitrogen bond of the two amino acids is broken by proteolases. And finally, I'm going to talk about acetylation, which is the addition of acyl groups to nitrogen on amino acid. It takes place when ribosomes is still translating the polypeptide chain. This also plays an important role in gene expression. The histones proteins that help condense DNA to chromatides can be acylated, which reduces its ability to fold and open DNA for transcription. So now we're moving on to protein degradation. Post-translational modification can also regulate the degradation of protein by ubiquitination. Ubiquitination is the ability of ubiquitin used as a marker for degradation. The purpose of protein degradation is to get rid of any damaged or misfolded proteins in the cell so everything can work properly. The amino acids also are recycled so they can be used to build a new functional protein that is needed. So two main mechanisms for protein degradation is ubiquitin and proteasome pathway and lysosome and proteolysis pathway. The three components of this ubiquitin pathway are ubiquitin activating enzyme, which is E1, ubiquitin conjugating enzyme E2, ubiquitin lysis, and ubiquitin lysis. So looking at the figure, ubiquitin is activated when it's attached to E1, and then ubiquitin is transferred from E1 to attach to E2. E2 with ubiquitin and E3 bind to the target protein. E3 covalently attaches ubiquitin to licensed amino acids on target proteins. The ubiquitin chain forms and E2 and E3 both dissociate. Then the target protein is then degraded by proteasomes into short proteins, peptides. So lastly, I want to talk about the lysosome proteolysis. It contains digestive enzymes such as proteases and hydrolytic enzymes that degrade surface proteins. In this pathway, proteins are enclosed in membrane vessels and fused with liposomes. Then proteins are broken down by hydrolytic enzymes. In this pathway, they are typically non-selective degradation of proteins, which can cause a slow degradation and to be a very slow process, and it can be harmful to the cell. So this concludes this group's presentation on regulation of gene expression. So thank you for listening.
So in this video, we were able to see the different levels of gene expression and how these levels could be regulated. And we were able to cover all these levels: the genome level, the transcription level, spoken about by me, RNA processing and nuclear export, which is the post-transcription levels, talked about by Malik, the translation level, talked about by Nate, and the post-translation level, talked about by Nakia. So thank you for watching, and join us next time for our final project video.